I'm going to start talking about Sebastian. He is an assistant professor of metabolism with the Animal and Dairy Science Department with UW-Madison. Sebastian's research focuses on understanding nutrient metabolism and the overall objective of maximizing nutrient efficiency for milk production. Sebastian's goal is to understand the regulation of mammary blood flow, nutrient uptake, and milk component synthesis as a means of maximizing nutrient demand by the mammary gland. He is also interested in nutritional and endocrine regulation of hepatic metabolism of energy and amino acids. Sebastian started his education program in Uruguay, and then he came to Virginia Tech, where he got his master's, PhD, and first postdoc. And then he started a postdoc program at UW-Madison, where he is now a full-time assistant professor. So I see the slide is up, balancing diets for energy and amino acids to maximize milk components. So I'll turn it over to you, Sebastian.
Thank you, Matt. Thank you for the introduction. Can you confirm that you can hear me well?
Yes, I hear you, Sebastian.
Perfect. And the slides are fine, correct?
Yes.
Great. Let's get started then. So yes, today I'm going to talk about the interaction between energy and amino acids as the new models, the new version of CNCPS is, I hear is coming, the NASEM is already available. That the interaction is more more relevant, or we seem to be know more about it and how to represent it. So it's important to understand that. Before I start, I want to thank the student that did the work. I was just here in the back of the terrace waiting for the results. Especially Catherine Ruth on the left and Marjorie Killerby, that are the ones that led the studies that I'm going to present today, but also the rest of the lab, because these studies are very intense and require a lot of help. There are some of the students here that some of them already graduated, but there is many more that I couldn't find a picture with all of them. And I also want to thank the funding sources, USDA, NIFA, and also USDA Hatch, and Adiseo, who fund some of the studies that I will present today as well. I want to start with some of the definitions that I'm going to be using today, so everyone is on the same page. Nitrogen use efficiency, amino acids, crude protein, metabolizable protein, milk-through protein, forage NDF are some of them. There are some others, hopefully they're obvious for you. So why nitrogen efficiency? The improving nitrogen efficiency has a three-way reasons. First, you know better than me, protein is the more expensive ingredient in your diet, in a lactation diet, and the diet is the more expensive cost in the production. So reducing nitrogen supply can definitely reduce the production cost and improve the financial sustainability of the system. Reducing nitrogen losses, improving nitrogen efficiency is also going to improve milk components production, and that is not only a reduction in the cost, but it's also going to be an increase in the income. But also there is an environmental reason to reduce nitrogen emissions, that although before we were talking about possible regulations, nowadays I think that most of us have heard about regulations regarding emissions not only of methane but also of nitrogen, nitrates in underground waters, ecosystems, water ecosystems. We are radiated by one of the most important sources of water in the world, that are the Great Lakes. So there has been, and you can hear in the news, it has gone to the Wisconsin Supreme Court. There are many reasons. The pressure of reducing nitrogen, and consumer pressure as well is increasing. So it's important to at least have the tools, not like how it's happening in the Netherlands, for instance, that producers are having to sell cows, reduce their stock because there are no tools to reduce nitrogen emissions to the level that the government is requiring there. And so hopefully we have the tools by the time that the regulations come. Amino acid supplementation is something that I'm pretty sure that most people have heard about this. It's more common. We start talking about crude protein. More recently, we start talking about metabolizable protein. But nowadays, I think it's fairly common to talk about amino acid supplementation, specific amino acid supplementation. And I'm going to talk about why it's important to balance for a specific amino acid, not just protein, for which amino acids, and a little bit of how much of those amino acids we should be considering. I'm also going to talk about different energy sources, forages, starch sources, non-forage NDF sources like soy hulls, citrus pulp, beet pulp, and oil fats that Lisa did a great introduction to the topic. So that's going to get us well prepared. And finally, I'm going to talk about the integration of or the mixing of the energy sources and amino acids. I don't think I'm going to have a lot of time to talk about the rumen, the liver, and the mammary glands, but overall production, I'm going to be showing some results from our group. So to get started, how much nitrogen does a lactating cow waste? Well, these are different meta-analyses and reviews that one is done by us in 2014. There is another from SPEC in 2013 and more recent in 2020. In this case, it was very comprehensive because include organic and conventional and jerseys and non-jersey cows. So it's fairly comprehensive, and despite it was almost 10 years later, we are in the same about 25 to 30% nitrogen efficiency. So there hasn't been much progress in that regard. So it means I mentioned before that protein was the most expensive ingredient in dairy cow, lactating dairy cow diet. And we're only recovering between a quarter and a third of the nitrogen that we are feeding that cow. So that means that there is a lot of money that is going down the drain. So for instance, this is from a paper that just came out a couple of weeks ago in Journal of Daily Science, and I'm just using it as an example. A cow that is being fed, the positive control was being fed a 17% crude protein diet, fairly common. That cow was consuming 48 pounds per day and was producing 83 pounds per day. and so where are the losses going? About 30% of the losses are going in feces, 25% of the losses of the nitrogen consumed is going in urine. And in this case, there was some protein acquisition, so it's a little bit larger. Otherwise, scarf and other small losses are typically below 10%. And in this specific example, with a 17% crude protein, the cows were recovering only 28% of the nitrogen consumed in milk. So the most important losses are because of they are the larger and also they are the ones that we have more capacity to manipulate, are the urinary losses and the fecal nitrogen losses. The fecal nitrogen losses consist of indigested proteins from the diet, from the microbes that ferment the diet, and endogenous losses. But it's important to keep in mind that these are indigested proteins. So this is a form of nitrogen that is gonna take time to degrade and is not likely to cause the environmental impact that other losses that I'm going to talk about cause. These are losses that some way when you spread the manure and to fertilize the soil, the crops, it's slowly going to degrade. It's going to be incorporated into the soil, and plants are going to be able to use it. So the losses of the nitrogen are out of the system are fairly low. There is also in physics some recycled urea. It's hard to quantify, but this is, but most of the urea is going to be excreted in urine. And where is that urea coming from? There are two sources of urea in urine. One of the sources is ammonia that was absorbed from the rumen. And that happens when we feed high RDP diets. The microbes don't have the energy and don't have the time to capture all the nitrogen that's in the rumen. And so that nitrogen is being removed, goes to the liver, and is converted to urea, so the ammonia in blood doesn't cause toxicity. And that urea is being excreted mainly in urine. Also, you can measure that urea in milk. The other is the catabolic losses. That is, and this is even more concerning because it has the much higher cost. RDP is less expensive, but bypass proteins are the most expensive. And these are proteins that we fed the cow, they escape the rumen, so these are bypass proteins, or they were converted to microbial amino acids, and they were absorbed by the cow from the small intestine. They were not utilized by the mammary gland. Is the main use of amino acids in a mature lactating cow. Is going to be for milk proteins. So these were not utilized by the mammary gland, and they return to the liver. The liver say, okay, this is too much amino acids. We have to clear a little bit, and it's going to be converting it to urea, while the carbons of those amino acids are being used for glucose synthesis mainly. The urea, it's much more concerning because the urea can be rapidly lost. Unlike the indigested proteins, urea can be lost in by evaporation as ammonia. It can leach down to underground waters as nitrate, or it can be through the nitrification process, it can be excreted as nitrous oxide, which is very potent and long-lasting global warming gas. So these are actually the losses that we should try to minimize. We should try to pay more attention, but also these are the losses that we can do something with dietary strategies. The indigestible proteins are very hard to reduce that part, and it's more with industrial technology, with engineering of those proteins, industrial process like preventing heating of the proteins. So getting a high quality protein, high quality bypass protein, we're gonna reduce the fecal losses. But it's through correct diet formulation that we are gonna reduce the losses of urinary urea from the rumen and from catabolism. So if we open this, the ammonia, the recycled amino acids, and the indigested proteins, these are the three main losses. We already said that ammonia and recycled amino acids are the most concerning ones. So if we go back to this example in the paper from Choudhury that was recently published, they also had a negative control treatment, a low crude protein diet that reduced from 17% to 15% crude protein. There was a numerical decrease in dry matter intake. The number of cows was small here. In other studies, this difference of about 2 pounds can be detected, but in this case, it wasn't significant. And there was also a decrease in milk production, but it was only numerical. They didn't detect it. So there is, importantly, there is between the two studies, there is a 25% increase in nitrogen efficiency when we go from considering the changes in dry matter intake and in the reduction in crude protein and the output in milk. There is a 25% increase in nitrogen efficiency, relative increases in more stable fecal nitrogen. So the one that is being, the nitrogen that is being reduced, the excretion is the more harmful one, the one in urine. So the proportion of the total nitrogen excreted, that is fecal nitrogen, is larger, which that is good. And because of that, there is an absolute and relative decrease in urea nitrogen losses. I had a diagram here to show that, but it didn't, it's not showing up, I'm not sure. So the catabolic, we already talked a little bit about the rumen. I'm gonna go back with some of the data that we have to the rumen, but I'm gonna focus more on the catabolic losses, that's where we have been working the most. That is the losses of metabolizable protein. Metabolizable protein is the pool of amino acids that were digested in the small intestine and were absorbed. So ideally, they are now secure. But we see here that, and this is a review paper from Dr. Lapierre in 2012, that the more metabolizable protein supply that we provide to the cow, the more production there is. But this increase in production in response to metabolizable protein supply is not linear. There is a positive diminishing response. So there is a positive response to metabolizable protein supply, but it diminishes as the level of metabolizable protein increases. So that means that we start with an RDP that is fairly inexpensive, fairly cheap, and we get to about 1,000 grams, let's say. But then when we start supplementing with the bypass proteins is when our marginal efficiencies start to decrease, our diminishing responses start to happen. So the more that we are spending in high quality protein sources, the less that we are getting. And that's why we are going to talk about balancing for amino acids to try to avoid the drop in the response. One thing is we have to keep in mind is supplementing amino acids versus substituting the protein or balancing, because when we are supplementing on top of what we are already feeding, we are increasing the metabolizable protein supply, and it's likely that we are going to reduce that efficiency of nitrogen utilization. Meanwhile, when we substitute, when we just balance what we are already feeding, when we correct without increasing the MP supply, we are not gonna sacrifice efficiency, and so we are not gonna increase the losses of that more expensive last portion of nitrogen being supplemented. So in this case, this is a study, a beautiful study done by Hake and collaborators in France from Dr. Lemosquetti group. And so what they did is they infused the amino acids in the abomasum and the true stomach, but they have 3 treatments. They have a treatment in which they infuse just I think it was glutamine and glycine, so non-essential amino acids. They infuse the same amount of nitrogen, but as histidine, lysine, leucine, and methionine for essential amino acids, for limiting amino acids. And then they, in the third treatment, they infuse all the 10 essential amino acids. And they did this at a low protein level and a high protein level. So what we see in terms of true protein content in milk and true protein yield is that first, when we increase from low protein to high protein, in both cases, there is a milk protein response. I already showed you in the previous graph in the meta-analysis done by Lapierre that true protein yield responses to metabolizable protein supply, but the responses in a diminishing way. So the benefit keeps reducing. But what is more interesting in here is that this, in addition to this response to the increase in metabolizable protein supply, we see an increase for the type of amino acid between the control, the 4 amino acids, and the 10 essential amino acids. There is no difference at each level of metabolizable protein. There is no difference in nitrogen supply, in protein supply. It's exactly the same. The difference is what type of amino acid we are providing. And what we see here, especially in the true protein yield, is the one that we should care the most because it's based on what the check is based off, is that the true protein yield is not different when we only balance for 4 amino acids versus when we balance for 10 amino acids, that it's going to be much more difficult to do and it's going to be more expensive. In both cases, we see a response compared to providing just non-essential amino acids in the supplement, but the response is comparable. It's basically the same when we substitute those non-essential amino acids for these 4 essential amino acids, histidine, lysine, leucine, and methionine, or when we substitute it for the 10 essential amino acids. That is the difference between supplementing, that is from the low protein to high protein, to substituting or balancing, that is changing what type of amino acid without increasing nitrogen supply. So which amino acids should we balance in for? These 4 amino acids, Hake already showed that there was a response, and I added some meta-analysis. This is not meaning to show all the data that is published. There is, in the last 30 years, there has been a huge amount of data, a large number of studies on individual amino acids or group amino acid supplementation that it would be impossible to cover here. But I'm trying just to summarize it with some of the meta-analysis. These are the 4 that I'm aware, just the 4 amino acids that have shown responses in meta-analysis: methionine, lysine, leucine, and histidine. In this case, there are several meta-analyses for methionine and lysine because, of course, we have been supplementing with those amino acids for a long time, and it's the only 2 that I'm aware there are commercially available products. There are some prototypes for histidine, and there are several studies using those prototypes on using abomasal infusions of histidine as well. And last year, Susanna Reisenen, from Dr. Histro group, published this really nice meta-analysis showing a very interesting response to histidine as well. This is a meta-analysis from Lin and collaborators that shows a response to leucine as well. And there are some independent studies that have shown response to leucine, but these 3, methionine, lysine, and histidine, are typically recognized as the most limiting ones. But I'm going to show you some ways in which we could balance for leucine as well. Isoleucine is another amino acid that is considered by NASEM, the new nutrient requirement system from NRC, but there is no meta-analysis for isoleucine on the milk protein responses. There is a meta-analysis from Versiana, Daly, and Dr. Hanian that shows a positive effect of isoleucine on milk fat production, which is very interesting as well. And then there is sporadic evidence for other amino acids, but it's very limited and it's really hard to cover all that. So I just focus on the ones that are considered now by the NASEM model and that we have a fairly strong evidence regarding the response. So a big change in the new nutrient requirement system is that now we have independent linear responses to these 5 amino acids. What means independent linear responses? It means that we were used to the concept of the barrel to represent amino acid balance. And if there is one amino acid that is most limiting, it's not gonna matter if I supplement other amino acids because the cow cannot respond. Well, we show that in several studies now that that is not the case. And that is the cow is short in methionine, but it's also short in leucine. We can supplement leucine and the cow will respond regardless of the level of methionine. And those responses are going to be additive. If we add the methionine on top, the cow is going to respond more. Those responses are still diminishing. So the more that we add either of metabolizable protein or metabolizable amino acids are going to decrease. So you have to pick depending on the cost of the source of nitrogen that you are supplementing, where you want to be, if it's in a in a low level with a high response, with a very steep response, or if you want to be more towards the plateau of the curve with a more shallow response. But that is going to be a combination of nutrition and economics of the product that you're using. There is also effect of other amino acids. And there is, as I mentioned at the beginning of the presentation, an effect of energy. So what the model doesn't consider is what type of energy. And another thing that the model doesn't consider is an effect of amino acids on fat or lactose, on other milk components, which I think that we have some results that are very interesting in that regard. But I remember a professor told me once, calories are just calories when I was in my PhD. And then when I did my postdoc, Matt mentioned I did my postdoc here at UW-Madison, but I did it in the School of Medicine. And that wasn't the idea, what I understood, especially with human application, that calories are just calories. It's not the same if we eat carbohydrates, fats, or protein. So that's something that I wanted to investigate a little bit more. If that is the case, if all the calories are the same for the cow, as we know, it's not the case for us. And so, what are the main energy sources in a lactation diet? We're going to have forages which are mostly ketogenic. That means that the fiber fermentation, the products of the fiber fermentation, the cow cannot use them to produce glucose because it's low in propionate. And the overall problem that we have with forages is the rumen feeding effect. We also have starch sources that are mostly glucogenic because that is the main product is going to be propionate. But we have two risks. One is the risk of acidosis, and the other is the risk of milk fat depression that Elisa briefly mentioned. If we combine, especially if we combine high levels of starch with some specific fatty acids. But then we have this other group of ingredients that are less common in lactation diets, but now as the availability of byproducts increase, we are starting to see more and more, but we still don't know much about them, how to combine them. And one is the non-PNDF or non-forage NDF, like soy hulls, like citrus pulp, like beet pulp, like almond hulls. If you go to California, you're going to have much more of those type of ingredients. They are similar to the fiber. They are going to produce low amounts of propionates, so they are not going to be glucogenic. They are going to be ketogenic, but that also means that they are not going to are not at the risk of acidosis that high levels of starch, and they have high degradability and high passage rate. And finally, oil and fats that by now I hope you already know quite a lot about them. We also have protein. Protein is a source of energy. We can, when the cow produces that urea, can combust the carbon and obtain energy. But hopefully, we are not using that protein as a source of energy. Hopefully, that protein is being used by the cow as a source of amino acids for synthesize her own proteins, her milk proteins, her liver proteins, her blood proteins, uh, all the proteins that the cow is going to need for to meet her basic functions and production. So I talk about different sources of energy, and we know that if we substitute starch for NDF, for forage NDF, there is likely going to be a decrease in production unless we are in a very high level of starch that is affecting rumen function. And that is shown in this study published in 2016, sorry, in which they have 4 different ratios between NDF and starch. They have from, and they did that substituting corn grain with corn silage and oat hay. So the level of crude protein is all the same, but the level of NDF increased from treatment 1 to treatment 4 as the level of starch decreased from a very high level, 34%, to a very low level of 17%. I would say that the T2 and the T3 would be more in the range of commercial diets. Now, what happened when we substitute this starch with NDF? We see with forage NDF, we see that there is a drop in energy content of the diet. There is a drop in dry matter intake because of the filling effect of NDF. And there is also a drop in the metabolizable protein supply, because if the rumen is working less, there is going to be less growth of microbes, and there is going to be less metabolizable protein available for the cow. And in line with those changes, we see a significant drop in milk production, significant drop in milk fat production, despite forages are help milk fat because they produce the acetate. There is still, with less energy available, there is less milk fat production, and there is less milk protein production. So just substituting forage NDF with starch will increase the energy content, the dry matter intake, microbial growth. With that microbial growth, microbial activity, BFA production, microbial protein passage to the small intestine, and with that MP supply. And so with more BFAs and more metabolizable protein, the cow is going to produce more milk contents as well. That is not new. That's something is familiar. Likely. But what happens when we use sources of NDF that are non-forage NDF, that don't have the filling effect, that they don't depress dry matter? And then is the advantage of the of starch the same? Well, in this study from Dr. Shaver group, Randy Shaver group, I think Luis is one of the authors that it was there. There are few cows, because they were all cannulated cows. They have increasing levels of corn silage or increases levels of soy hulls, while they decreases the level of corn grain. And so they end up with no changes in dry matter intake and not significant changes in milk production either. When we look at fat and the protein, there are no changes in milk contents either. So when we use a non-forage NDF source, we don't see and we don't have an effect on milk on dry matter intake. We don't see the responses in milk components, the benefits of using starch that we see when we are sacrificing, when we are compromising rumen capacity and dry matter intake. And so the evidence supports balancing for the 5 amino acids. And also the evidence show that we could reduce the amount of starch if the price or the availability is limited, or if we don't wanna risk acidosis, or if we don't wanna risk milk fat content, we can substitute for starch without necessarily compromising milk components. The question that I wanted to address that I haven't seen it before being addressed is if the amino acid supplementation depends on the type of energy that I have in the diet. It's the same if I supplement with these 5 amino acids or with some of these 5 amino acids when I have a high starch diet, that when I have a high NDF, or also when I have high fat in the diet. Are the amino acids going to respond the same? Are all the calories the same for the response of milk protein? That's something that we don't have very clear answers yet, and I decided to explore. So in the first experiment, in one of the first experiments, we did several. We did in vitro work. We did some work with mice, and then we did some infusions. But I'm going to show you the diets because I think that's the most relevant. Those type of studies help me to establish the hypothesis stronger. But we need to. The cow is the one that is going to say the truth. So I'm going to show you 2 studies in which we use dietary approaches, not infusions. And in this study, we use a total of 6 diets, 3 different energy sources, combination of energy sources. Corn silage was always the same, but we decrease the amount of alfalfa silage from and the amount of corn grains. So we have the first diet is a high starch, high forage NDF, because we need the forage NDF to avoid rumen problems. We also have a high starch, high fat diet in which the forage NDF, we use more straw because they were isocaloric, the diets. And then we have a third diet that it was low starch, and we use the same high fat and high non-forage NDF because we don't need so much of the forage NDF to buffer the rumen when we don't have the starch. So that is going to give us some more space. And for the fat, we use an 80% palmitate and 10% oleic acid. It's a commercial product. Then within each energy source, we use soybean meal as the main protein, the basic bypass protein source. And to balance for methionine and leucine, 2 of the amino acids that NASEMs consider that have direct effect on milk protein, we added corn gluten meal and we added rumen protecting methionine. So in this case, we are adding to the basic diet that it was about 10% protein deficient. So at the end, we have basically 2 levels of NDF. The first 2 diets have the same NDF level, and the last diet has higher NDF level, but because it's non-forage NDF, we have 2 levels of forage NDF, 28%, which is very high, and 17%, which is very low as well, and 2 levels of starch, 30% and 23%, and 2 levels of fat as well, 2.5, that is just the fat content of the diet, or about 5% with the supplement. And the idea was the RDP to be all basically the same, although there were some variations. But the metabolizable protein was the same in the three energy sources and either supplemented or not. So the first thing that we have is, okay, if we, when we were supplementing, substituting starch for forage NDF, we have a depression in dry marine intake. Now, when we are substituting starch with non-forage NDF, as we don't need the forage NDF to balance the rumen pH, we can supply more, and we actually see here a bump in dry marine intake. And in part because of the response in dry marine intake, and in part because of the sources, perhaps, we also see that the low starch diet, 23% starch diet, low forage NDF, only 17% forage NDF, and high levels of non-forage NDF and fat supplementation, we see significant increases in milk production, in milk components production, and we see that despite because of these increases, there is a decrease in milk urea nitrogen. We also see that amino acid supplementation increase milk protein yield and milk fat yield. But in this case, that it was an addition, we also see that amino acid supplementation increased milk urea nitrogen significantly. Now, okay, in this case, we have a response in dry marine intake. That's a good strategy. We can do it, but that doesn't answer the question of the interaction between energy and amino acid, because we have the confounding factor of dry marine intake. So in the last experiment that I'm gonna show you, what we did is we fixed the forages. All the forages were at the same amount, trying to have the same level of dry matter intake. So the corn silage and alfalfa haylage were the same. And then we have the different energy sources. We have a high level of starch with corn grain. This is dry corn grain, or a low level of starch supplemented with the same soy hulls and the same fat source. And we aim for the same metabolizable protein, but because, as you will see, the models don't do a very good job predicting the fermentation of the non-forage NDF sources, highly degradable, the model was telling us that we have to have the same metabolizable protein, we have to supplement more protein in the low starch diets. So that's why we have more crude protein, but based on the model prediction, both Cornell and NRC, the same MP level. And then in each of the energy sources, we supplemented with, we balanced for leucine, lysine, and methionine using rumen protected sources and corn gluten meal. That is very high in leucine, has about 20% leucine. The composition of the diets end up being all with the same RDP. That was very important for us, so we can use the same for the forages in the same way. We have basically the same metabolizable proteins that increase when we balance for the amino acids, of course. And we have basically two levels of NDF, two levels of starch, 28% and 20%. In this case, we were even more aggressive than in the previous study, and two levels of fat, 3.4% and 5.6%. So first of all, good job. We don't see an effect of energy on dry matter intake. Between the high starch and the low starch, we have exactly the same dry matter intake, so we can guarantee that that the effects are more because of the energy source and not because of change in dry matter intake. We do see an effect of amino acids on dry matter intake, and contradicting our hypothesis that the different energy sources could affect the response to amino acid, we don't see any interaction between the factors. There is an effect of energy source on meal components, and there is an effect of amino acids on meal components, but they respond independently. So what do we see in terms of energy? Something very similar to what we saw in the previous study, where there were differences in dry matter intake, although here we don't have them. Switching from a high starch diet to a low starch diet with soy foods and fat, isocaloric, we see an increase in meal production. We see an increase in energy corrected milk of about 8 pounds, 4 kilograms. We see a very significant increase in milk fat yield that if you compare the two control treatments is about 200 grams, so almost half a pound. And we see an increase in milk protein yield as well, smaller but very significant as well. When we supplement with a balanced amino acid mix, trying to supplement the levels just of the amino acids that we are targeting, we do see an increase in dry matter intake. We also see an increase in milk production in energy corrected milk. In energy corrected milk is also about 6-7 pounds. We see an effect of milk protein, which was expected. The effect of milk in the increase in milk protein, you can see that is about 150 grams, depending on the treatment. But we also see a very strong effect on milk fat. Of course, not as strong as the energy source, but there is about 80 grams of milk fat response. So if you try to do, we don't have the time now, but you can do the numbers with the costs that Lisa gave you. And I think that this is going to be very profitable responses of using fat sources as well. I also want to show you this. We did some modeling work. We got the NASEM, we got the raw code of the NASEM, and we ran individual cows to see what happened. And also we measure microbial protein. So using both the NASEM model and measuring allantoin in urine, we see that there is an increase in microbial protein passage in the low starch diet. And this suggests that the models, as I mentioned before, were not doing a very good job predicting how much the microbes can use the soy foods, okay? And because there is an excess of protein, we provide more protein because of this, because of what the model was telling us, we see that there is also an increase in milk urea nitrogen, increase in urine in response to the low starch diet. Now, the last thing that I want to highlight is that when we start balancing for amino acids, when we start adjusting, tweaking the diets and giving a more balanced diet, the urine losses that used to be much larger because we were dumping a lot of nitrogen in the rumen, because we were feeding excess nitrogen as well, 19% crude proteins. Now, the harmful nitrogen starts to be a much smaller proportion of the total nitrogen excreted by the cow compared with the fecal nitrogen, which is more stable, and we have less tools. So here we have tools to really reduce the harmful nitrogen and improve the production and reduce production costs. So in conclusion, and I think a little bit over time, energy source does not affect the response to amino acid supplementation. So there is no interaction. You're going to have a response to energy source. You're going to have a response to amino acid supplementation, but those are going to be independent. However, reducing starch without affecting dry matter intake could increase milk fat. And we also saw an effect in milk protein yield. So that's something to keep in mind, especially with milk fat, with the current prices of the different components. And it's also to keep in mind, it's also very important that the models, doesn't matter if it's CNCPS or if it's NASEM, they don't do a really good job predicting the responses to this type of supplements. If you tell the model that you're going to use soy foods, that you're going to use fat, and that you're going to feed only 20% starch, the model is going to tell you a different story than what we see in the cows. And finally, supplementation with balanced amino acid, with this target amino acid that NASEM is focusing on, increases meal component production, including fat, something that is not represented by the model, without significant increase in nitrogen losses, which is what we really want to do. So I think that it's something that is showing to be really worth starting to pay attention to the different amino acids. And not in every case we have to invest in very high protein very expensive protein sources, or in rumen protected amino acids. There is some cases that is going to make sense, but there is other cases that using a blend of different proteins, we can balance for, at least partially balance for some of them. I didn't show any results of histidine, but soon we're going to have also some very interesting results of histidine. So with that, I'm going to pass it to Matt, who is going to remind you about the next talks, and I would be happy to answer any question.
Yeah, okay. So before we get to the next, well, I guess we can do it, it's up there. Our next Badger Dairy Insight will be on March 19th, and it'll be Paul Fricke's reproduction group, and also J.P. Martin's large animal, it's a reproduction day, strategies for improving reproduction in dairy herds. If you go to the Badger Dairy Insight website, you will see that we are starting to populate this with, you can access recordings. So especially, Sebastian, what you put out there, I mean, you had really big p-values or tiny p-values, I guess that'd be the better way to say it, and a lot of things going on, starch and amino acids and protected fats and soy hulls. So I think a lot of really current topics in the industry. When that gets recorded, I'm going to want to come and look at that again, because even though you gave a thorough presentation on it, I need to see it again. So just so you know that those things will be available. If not immediately, you can come back to them. And I see that we have a couple, they showed up as a Q&A instead of chats. Francisco says, excellent talk, Sebastian, congratulations. And are the recent studies already published?
First, thank you so much, Francisco. And they're going to be published hopefully very soon. If my students are looking at the presentation, they should be looking at working on the papers, because we're almost done. But we already published the production responses in ADSA in the annual meeting. So if you go to the Journal of Dairy Science, you can find those abstracts with the production responses. The authors are Catherine Ruh, R-U-H. I'm the last author in all of them, and Marjorie Killerby is going to be the last. I was thinking because Marjorie has been working in that study, but we started before she joined. But no, she's going to be the first author in the other study. So you can find that information. And Marjorie is going to be presenting a study we just completed on the use of histidine in ADSA this year as well. So that, and the papers, as I said, we are hoping to send both studies, the papers, by the end of the month. So you just pay attention to the Journal of Dairy Science, that if not rejected, we are going to have them published soon.
Okay, thank you. And then this other one came up during Liz's presentation, and our panelists, we've been talking about this a little bit. So Hussein is asking, what's the recommendations regarding bypass fat 84? And I think that's referring to a calcium salts type product. And then products that are more pure fat, 99%. So I know that there's quite a range of products available in some of those classes, but he's asking, especially in summer, so heat and high yielding animals. And I see Lissa's there, but Luis, you want to, we conned you into talking about this too. So, you know, I know that some of the high producing cows on farms are getting fed both in blends. And you know, all the detail that's going in these really good rations nowadays, sometimes it just can be deceptive that people don't just account for the difference of fat in the cost, which obviously is a very basic thing. But I'll leave it there. And Lissa muted herself. So Luis, you're the one.
Sure, yeah, I can start to answer that. And please, Sebastian, Lisa, feel free to chime in. You know, it's I think that the 99% protected fat is based primarily on palmitic acid, right? The C16. And some of the research associated with that suggests that, especially during the summer, if cows are fed with C16, there is potential to increase milk fat. During the summertime, I think this is quite important because due to some of the issues associated with heat stress, there are a lot of mineral losses through saliva that cows undergo. So rumen pH goes down, and then if you have a lot of unsaturated fats, you may end up with this scenario of milk fat depression that Lisa discussed earlier today. You know, so it could be a good option related to that, right? Obviously, in addition to that, you have to pay attention to all the other ingredients in your diet, strategies for heat stress abatement, adding different buffers when possible, and pay close attention to sorting, right? To make sure cows eat their forages and have good rumination to assist with that, right? And again, Lisa, Sebastian, please feel free to chime in.
Yeah, it's just very briefly, I totally agree with what Luis said. And as I typically teach in my ruminant nutrition class, as I show in my studies, you're gonna have about 3.5% ether extra in your diet based just on the forages and the grain that you use. And then if you use some protein source that has oils, you want to make sure that that doesn't go beyond another 1%, because otherwise you can have problems of milk fat depression. And then you can add between 1.5%, 2% more. So getting to 6.5%, even 7% nowadays with rumen inert or rumen protected sources is still safe. And as Luis says, in the summer it's really good because it also, those fat sources don't ferment, so they don't produce heat and can help to reduce the heat stress.
Okay, so we've exceeded our time. But I just had one I wanted to ask Sebastian. When you were kind of setting the table and talking about the need for efficiency, and you talked about the Netherlands and having to depopulate, do you have a sense how important is tweaking in the diet of the cow? How much, let's do nitrogen, how much relevant that is compared to the whole production system when we're growing this corn silage and we're putting all this nitrogen, or we're growing grasses and we're putting all this nitrogen? Is what's happening in the cow nearly as big a factor as what's happening in the cropping system?
I'm worried to not tell you the correct answer. That's an excellent question, and it's something that I have been looking for the correct answer for a long time. I have talked to people that work more like a system level and modeling. I think I only saw one research paper that said that actually, if you reducing the cow, then it has more impact than the losses in the manure and the losses in the field. But don't believe me, because it's something that I'm not sure about it, and I think it's really critical to research on more like a system level, what is going to have the most impact to really try to address the with less input to try to have more impact in the reduction of nitrogen. But yeah, I'm not sure about that. I don't know if you, Luis, know those numbers.
Yeah, I'm not familiar with those numbers, but I agree with your answer, and it's definitely something that we have to continue to pursue and understand. And at the end of the day, more and more we'll have pressure to mitigate any sort of extra inputs that we are feeding to the cow or excreting. So there's a lot of need out there to understand those.
The only very brief thing that I would add is is the problem that sometimes we have is that the system losses are many times predicted with large-scale models. And for instance, Dr. Histrov has shown that those models not always match what we measure in the cow, for instance, in terms of methane. So when people do the predictions of the impact of agriculture just based on 10,000 feet models with a lot of assumptions, those numbers can be really off.
Very good. So I am excited. It seems to me that today, if people want to manipulate fat or protein tests in their milk or muns, there's ways to do it. So thank you, our entire panel. And again, a reminder to come back March 19th. And with that, we've overextended our stay by a bit, 17 minutes. So thank you very much.